I'm an emeritus professor at the University of Utrecht. I worked at EMBL in the early 80s with Graham Warren, had a great time, very inspiring. And actually, we had our first couple of publications together with Graham, one in Nature, one in Nucleic Acids Research, which was a popular journal at the time. And so today I have assembled some slides as a broad introduction. For some of you, this might not be so much necessary, but last time I had the impression that many people were not too familiar with coronaviruses, so I will present this brief overview. It will be give some general features, go into the virion, the infection cycle, and more specifically into the way the virus enters cell, which is an important phase in the infection where people try to intervene, and also into the accessory genes. All right. Let me see. I want to shift. How to shift to the next slide? Yes. Okay. So coronaviruses consist of a big family of viruses, which initially were imported mostly in the context of the veterinary scene. And there on this slide, you see a number of coronaviruses that were and still are important in the field, in the veterinary field, like viruses in felines, in canines, in chickens, in pigs. And say before the first SARS coronavirus outbreak in 2002, there were 4 human coronaviruses known, and all of these were just causing mild common colds. So they were not considered very important. Well, by hindsight, from what we know now, but not at the time, we know that most of these, probably all of these human coronaviruses came from zoonotic events. We know that quite sure for the one at the top, human coronavirus OC43, which about 100 years ago jumped from cattle to humans. And that was historically, you can find back that there was a pandemic of bronchiolitis, so also quite severe symptoms, respiratory symptoms, as we know them nowadays. So already 100 years ago. So later on, we met with the SARS coronavirus in 2002, which started in southern China and traveled to Hong Kong, from where it spread to all over the world. As you remember, it was a virus that was coming from bats, but used civet cats most likely as an intermediate host. And then 10 years later, approximately, we had the MERS coronavirus that popped up in Saudi Arabia and was using dromedary camels as an intermediate host. And nowadays, of course, we have the SARS coronavirus 2. This is just an overview to show that these coronaviruses can be distinguished in different groups, different genera, alpha, beta, gamma, delta. And well, if you look in the middle, you see that they occur in many species. And in some species, you have multiple different coronaviruses. Like in pigs, you have at least 4. Also, the one at the bottom, the Delta coronavirus, which occurs in pigs. So these are quite multiple, and what they generally do is cause enteric and respiratory infections, which evolutionary is understandable, because those are the ways by which the viruses can spread and maintain themselves in the ecosystem through coughing, sneezing, and by the feces. They phylogenetically, they can be grouped together, and you find the alpha coronaviruses here in yellow, the alpha coronaviruses to which the 229E human coronavirus and NL63 human coronaviruses belong. And then you have here on the left-hand side different clades of beta coronaviruses, to which, for instance, MERS coronavirus here in this clade, SARS and SARS-like coronavirus in this clade, and here you have human coronavirus OC43 and mouse hepatitis virus, which I mentioned because it's the type member and was used to study coronaviruses fundamentally in most cases, certainly early. Okay, a little bit about the viral particle. It's coronaviruses are enveloped, which means that they have a membrane envelope, and they contain a positive strand RNA, which is the genome. And the genome size is extremely large. These are the largest messenger RNA-like RNAs which occur in nature. And if you look at the picture down here, the schematic drawing, which is which summarizes what we know of these viral particles. On the left hand, you see the typical EM structure of these particles. So in the viral particle, you have the RNA genome, which is packaged by one particular protein, multiple copies of that, to form a helical nucleocapsid, which is inside of a particle that is covered then by a lipid bilayer. And in this lipid bilayer, we find 3, sometimes 4 membrane proteins. The most conspicuous one is the spike protein, about which we will hear more. Then we have the M protein here, a triple spanning membrane protein it is, and then we have the E protein, which is an envelope, which is called envelope protein, but and that's what the E points at. So then below we see the genome structure. So this is the RNA with the 5' and a 3', typical messenger RNA, but very long. And about 2/3 of that RNA encodes the polymerase functions. It encodes a pol-1A and a pol-1AB, which are precursors that are subsequently cleaved into a total of 16 functional proteins. Downstream of the polymerase, we have the genes encoding the structural proteins. So we see there the S, the E, the M, and the N proteins, which are part of the viral particle. And in between those, you see, for this case, which is for a feline coronavirus, you see genes 3A, 3B, 3C, and 7A, 7B. And these genes are what we call accessory genes. They are in most cases, they can be deleted without the virus becoming non-viable, but they contribute specific functions, particularly in the pathogenesis. When we then look into a summary of the replication cycle of these viruses within a cell, so what you see is a diagram of a cell, and on top you see a virus particle binding to the cell by binding to a receptor, and then being endocytosed, and then the virus fuses its envelope, its membrane, with the envelope of the endosome, and that introduces then the viral RNA, the messenger RNA, into the cell. And this messenger RNA is seen by the cell as a normal messenger, and ribosomes start to translate it. And they translate from this long messenger only the pol-1A and pol-1AB part. So these 16 functional proteins that come from these precursors by cleavage, self-cleavage, most of them. They result in proteins that form structures in the cell that you can see here. We call them double membrane vesicles, and in which the virus hides to perform its transcription and replication. So we have there, on the one hand, replication within these structures, which means that the genomic RNA is by a polymerase that is generated, that is one of these 16 proteins that is generated from the POL1AB domain. It makes a copy, a minus sense copy that you can see here. This full strength minus sense copy then can be copied again and produce numerous plus sense copies of the original infecting RNA. And then what in addition occurs is synthesis of smaller messenger RNAs, which we call subgenomic messenger RNAs, from which the genes that are downstream of the polymerase, so the ones for the structural proteins, are then produced. And without going into the details, these messengers are then translated. They produce the spike, the envelope, and the membrane proteins, which are inserted into the endoplasmic reticulum, and which then are transported to the ERGIC, the ER to Golgi intermediate compartment, and that is the compartment where new viral particles are being assembled, because the full-size copies of the genomic RNA meet in the cytoplasmic the nucleocapsid proteins that are produced by one of these subgenomic RNAs, and they form this helical nucleocapsid, which docks to the membrane proteins, the viral membrane proteins that are accumulating in the ERGIC, and they then bud into the interior of the ERGIC, and this creates these vesicles which move to the Golgi and through the Golgi, and in the end the viral particle leaves the cell with fully mature glycoproteins, and the virus is then infectious and able to infect new cells. Okay, this is a picture just to show these double membrane vesicles, which these viruses use to hide and in order not to signal to the cells that there are foreign invaders, because cells are educated to react with all kinds of substances like interferons to counter the infection. So viruses have evolved to try and avoid those reactions from the cell. So here a picture where we see the process of virus budding. Here on the left you see the start of a budding particle. Here the budding is a little bit further. The blackened thing is the membrane, and you see it increase. Here it is almost complete, and here we have complete particles which then further mature and are released from the cell. How many particles are produced? Well, many, and that's interesting to realize, because, well, you can see here for the SARS coronavirus, the SARS coronavirus 1, this is after 15 hours in vero cells, and you can see here these vesicles with multiple viral particles, but also viral particles already on the outside of the cell, some of which rebind to the same cell, and most of them detach and go for other cells. This is at a very late point in the infection on the right-hand side, and you can see that hundreds and probably per cell more than thousands of new virus particles are produced, which then, if it is in the respiratory or nasal epithelium area, they find multiple new cells, and you can imagine that within a couple of days, a whole sheet of epithelial cells and these cells die and continue to produce new virus and then ultimately die. So the force of the infection is enormous. Then some more about coronavirus entry. This is again a picture of in the middle of the coronavirion, and on the right-hand side I summarize again the entry pathway with in yellow here the receptor, and this is then the endosome, etc. So it is the spike protein that is doing the entry job. And the spike protein is a normal type 1 membrane glycoprotein, which means it has a signal peptide at the N terminus, which is cleaved off, and it has a transmembrane domain at close to the C terminus, and after that a small endodomain. This is inside the viral particle, and the bigger part is outside of the viral particle. This spike protein can be distinguished. In that protein, you can distinguish an S1 and an S2 part, as we call it. The S1 part is the domain, more or less the top domain of the spike that is involved in receptor binding, and the lower part, the S2 part, is involved in the act of membrane fusion. Okay. The structure of this. Yes,
Peter. I think just because time is so short,
right,
and I really wanted to give a chance for people to come by and have some questions. I know we can do some of that in the follow-on, but right. could we? Yeah, if you could just maybe quickly
right.
wrap up this part. Yeah.
Right. So I will. Well, the spike protein is doing its job. It has it brings together the viral membrane and the host membrane. The spike protein the structure of it has been solved after many years. In 2016, we were the first to show the spike of the mouse hepatitis virus. And it's a really neat structure. If you can look at this, this is the surface view where you can see that the spike is a trimer. You see the three colors of the three molecules. They embrace each other very nicely, and you can see the different domains. You can see that in another type of view. And if you look at here in the middle, the top view, you can see that each spike falls into different substructures, and these substructures, domain A, domain B, for instance, they can function as the receptor binding domains. Here is one molecule here with the domain A and domain B at the left-hand side, and on the right-hand side you can see that for mouse hepatitis virus, domain A is the receptor binding domain. For other viruses, like for the SARS coronavirus as well as MERS, domain B binds to the receptor. What are the receptors that we know nowadays? Well, they are indicated here because here you have these different domains. For the NL63 human coronavirus, it is ACE2. ACE2 is also used by the SARS coronavirus, as we know, both the first SARS coronavirus as well as the one that is circulating now. Then we have APN, which is used by coronavirus 229E, but also by a porcine coronavirus. And then we have here DPP4 used by the MERS coronavirus. All these receptors are function normally as enzymes in the respiratory tract, and that is because probably the viruses use them because they are very abundant in the respiratory and enteric tracts. Okay, then a few words about the other genes, the accessory genes. This is again for the some different viruses, the genomes. The accessory genes, I could say that in summary, those are genes that usually interfere with the innate immune response of the infected cells. So they are trained to reduce the immune reaction by the infecting cells. Well, then I think...
Peter, I think we're going to have to move on just because of the incredible time pressure.
That's fine.
This is, I'm fascinated with this information. I'm sure, like myself, the audience has a large number of questions. And are you happy to take questions from the audience in the follow-up?
Sure, whenever.